This paper is focused in our work in the last years that is related mainly to the study of cell communication and clonal cooperation in tumor formation. In other papers, in previous papers, we demonstrated that cells lacking the integrin beta-3 are not able to give lung metastasis or in a significant less number of metastases. Then, in this paper, what we are trying to do, what we did, is just to explain why those cells with the integrin beta-3 don't give metastasis. And the main hypothesis was that probably is related to the exosomes uptake. And why the exosomes uptake? Because the exosomes carry a lot of microRNA peptides, cytokines, that are very important in cell communication, in the tumor cells, and with the environmental cells. Then, in our model right now, what we were able to demonstrate in this paper is that cells without the integrin beta-3 are not able, of the uptake of the exosomes is clearly reduced versus normal cells. And this probably is why those cells are not giving so much number of metastases. Then, because of that, we are working right now in the developing of new antibodies or peptides which block the integrin beta-3, because we are just planning to do in vivo experiments and why not clinical trials in the next future, because it's a way to prevent metastasis when we are able to inhibit or blocking cell communication because this new pathway.